At nearly 11,000 meters down, the pressure in the Mariana Trench soars to over 1,000 atmospheres, enough to crush steel, dissolve bone, and unravel biology itself in seconds. Yet, against all logic, life not only survives here, it's rewired the rules of biochemistry to do so. How do molecules and whole organisms adapt when every force in physics says they shouldn't exist? The answer changes everything we thought we knew about the possibilities for life, on Earth and beyond. At the bottom of the Mariana Trench, the numbers themselves defy belief. Challenger Deep plunges to 10,984 meters, nearly 7 miles beneath the surface. Here, every square inch is pressed by more than 15,000 pounds of force, 1,000 times greater than the weight of the air above sea level. That's the equivalent of stacking 50 jumbo jets on a single human spine. The pressure rises relentlessly, by 1 atmosphere for every 10 meters descended, until the water itself becomes a kind of slow-motion hydraulic press. Under these conditions, most metal hulls would be flattened like tinfoil. Even the most advanced submarines risk catastrophic failure. Yet in 1960, the bathyscaphe Trieste, piloted by Jacques Piccard and Don Walsh, made the first crewed descent to Challenger Deep. At nearly 11,000 meters, the vessel's thick steel sphere groaned under the load, but held. Through the tiny window, Piccard reported seeing a living creature, a flatfish, or perhaps a sea cucumber, stirring the sediment. It was a moment that shattered expectations. For decades, scientists believed life could not withstand such force. The Trieste's logbook, stamped with depth and pressure readings, became proof that biology can persist where the very laws of physics seem to break down. Here, the rules change. Proteins unravel, cell membranes buckle, and the familiar chemistry of life is pushed to its limits. The challenge is not just survival, but the fundamental re-engineering of life at the molecular level. Some organisms at the bottom of the Mariana Trench don't just endure the pressure, they depend on it. In the laboratory, a Hadal amphipod will die if brought to the surface and left at normal atmospheric pressure. Its biochemistry is wired for the crush. Dr. Anna Louise Reisenbach, a pioneer in deep-sea microbiology, has sequenced the genomes of piezophiles, microbes that require high pressure to survive. Her findings reveal a pattern. Their proteins are built differently. Compared to surface relatives, these proteins are enriched in small, flexible amino acids like glycine and alanine, which help the molecules bend, flex, and resist being squeezed flat. This flexibility is not random. A 2022 Nature study showed that these substitutions cluster in the most pressure-sensitive regions of proteins, reducing the risk of collapse at 1,000 atmospheres. But proteins alone aren't enough. Dr. Ken Y. Nagata's research at JAMSTech highlights a second line of defense: the cell membrane. Hadal fish and amphipods remodel their membranes with unsaturated fatty acids, molecules that stay fluid and functional where saturated fats would freeze or shatter. This keeps nutrients and signals flowing, even when the outside world is pressing in from all sides. Inside the cell, another adaptation takes center stage. Deep-sea animals accumulate trimethylamine N oxide, TMAO, in concentrations up to 3.5 molar, far higher than any surface fish. TMAO acts as a molecular shield, locking water molecules into stable networks and preventing pressure from distorting protein shapes. Without TMAO, the machinery of life would unravel. Yet, if these organisms are exposed to surface pressure, their chemistry fails. Proteins misfold, membranes leak, and death follows within minutes. For piezophiles, the trench isn't just a home, it's a requirement for existence. Below 1,000 meters, sunlight fades into complete darkness. In the Mariana Trench, life is cut off from the energy of the sun, forcing a radical rethinking of how food webs are powered. Instead of plants and algae harnessing light, The foundation here is chemical, not solar. At the base of this ecosystem, bacteria and archaea convert hydrogen sulfide, methane, and other chemicals seeping from the seafloor into energy, a process called chemosynthesis. These microbes use specialized pathways like sulfur oxidation, known as the SOx system, and methane utilization to fix carbon and build organic molecules, forming the primary food source for all higher life in the trench. In 2014, researchers discovered rich microbial mats near hydrothermal vents in the Mariana region, teeming with species that thrive on chemical energy. Dr. John Seawald, a geochemist who has studied these systems, describes these bacteria as the engines of the deep, generating biomass where no sunlight penetrates. Some of these chemosynthetic microbes cluster around seeps and vents, while others live buried in the sediment, quietly driving the carbon cycle in the coldest, dark reaches. Yet, chemical energy is not the only resource. A constant rain of marine snow, tiny particles of dead plankton, fecal pellets, and organic debris drifts down from the surface. This slow, steady fall of carbon sustains scavengers and filter feeders, providing a critical link between the sunlit ocean above and the Hadal depths below. Every scrap is recycled. Every molecule of carbon is used, making the trench one of the most efficient scavenging systems on the planet. Here, the food web is a delicate balance between the invisible work of chemosynthetic bacteria and the relentless hunger of organisms waiting for the next snowflake to fall. Water in the Mariana Trench hovers just above freezing, often between 1 to 4 degrees Celsius. This icy stillness slows every heartbeat and chemical reaction, forcing organisms to conserve energy and adapt their biochemistry to a world where time itself seems to crawl. Yet, only a short distance away, hydrothermal vents roar to life, blasting out superheated fluids at 400 degrees Celsius, hot enough to melt lead, but kept liquid by the immense pressure. Here, the boundary between freezing and boiling blurs, creating gradients that would destroy most known life. Dr. Julie Huber, who has spent years studying vent ecosystems, describes the Pompeii worm as one of nature's greatest paradoxes. This animal clings to the sides of vent chimneys, its head bathed in water as hot as 80 degrees Celsius, while its tail rests in a much cooler 20-degree current. The worm's survival hinges on heat shock proteins, molecular chaperones that prevent its own enzymes from unraveling in the heat. Specialized hemoglobins move oxygen efficiently across these temperature extremes, keeping the animal alive where most would perish in seconds. In the coldest stretches of the trench, other creatures fight a different battle. Their cells produce antifreeze proteins, which bind to ice crystals and prevent them from growing, allowing life to persist just above the freezing point. Enzymes in these organisms are fine-tuned to catalyze reactions at temperatures where chemical processes nearly stop. Each adaptation, whether for scalding heat or numbing cold, is a testament to the range of solutions life has engineered to survive at the very edge of what is physically possible. Snailfish glide through the Hadal zone, their bodies almost ghost-like, transparent, boneless, and soft as jelly. Instead of rigid skeletons, they rely on cartilage that bends with the pressure, avoiding the fractures that would shatter bones at 11,000 meters. Swim bladders are absent, replaced by tissues that neither compress nor burst. Amphipods, discovered in 2019 at Challenger Deep, reach sizes their shallow cousins never achieve, some stretching nearly 10 centimeters, gigantic in the world of crustaceans. These scavengers sport armored exoskeletons laced with unique minerals, adapted for the crush. Bioluminescent organs flicker in the darkness, sending coded flashes across the gloom. Dr. Robert Ballard, who has witnessed these forms on deep dives, describes them as life from another planet. Each body plan is a living answer to the impossible: how to move, feed, and signal under 1,000 atmospheres of force, where the ordinary rules of biology no longer apply. DNA in the Mariana Trench faces a relentless assault. Under pressures exceeding 1,000 atmospheres, molecular bonds stretch and snap, accelerating damage far beyond what surface life endures. Yet, trench organisms are armed with an arsenal of repair enzymes. A 2021 science study revealed that deep-sea microbes upregulate reshaping, a protein that orchestrates the repair of shattered DNA strands. This molecular first responder patches breaks before they become fatal, allowing life to persist where mutation rates soar. Dr. Chris McKay of NASA points to these adaptations as a blueprint for survival in alien worlds. Europa's subsurface ocean, locked beneath kilometers of ice, may mirror the trench's darkness and pressure. If life exists there, it will need the same genetic armor found in Hadal microbes. Already, industrial labs are testing trench-derived enzymes for use in biotechnology, seeking catalysts that work where ordinary proteins fail. The trench, once seen as a final frontier, now offers clues to life's reach across the cosmos and new tools for science on Earth. Right now, scientists are mining the trench for pressure-proof enzymes and clues to life beyond Earth. Each discovery rewrites our understanding of where biology ends and possibility begins. The limits we imagine for life aren't boundaries. They're invitations to look deeper. What boundary would you challenge next?